And good morning, good morning, good morning, good morning. Welcome and thank you for finding. My son Jack and I were looking for egg last night and we didn't find it. And it was one of the very first ones that you folks got. So well done. Let me take these off. Don't lose your annotation tools. We're going to use them again, of course, throughout the today. So this is our week of life history strategies, right? That's where we are. We were talking last week about just some shared lexicon and understanding that some of us have been introduced to some of these theories before, but now we have an opportunity to bring everybody up to kind of the same level and also emphasize some invertebrate examples because of the biases that we tend to have in how we educate people in biology, where we focus on the vertebrates, the things that we know. The Dothraki, that it is known, as opposed to the north of the wall Jon Snow, it is not known. And we'll talk about that, the context of it is not known, or Jon Snow, you know nothing, today. So we introduced some life history parameters and life history strategies. We talk about some different analogies, whether it's a piece of pie or a team's salary cap. We talked about different kinds of reproduction, which is where we're going to pick up the threads today. Because today we're going to talk more about the pros and cons and the different selective pressures that might have led to these different kinds of sexual reproduction, right into whether or not you are gonochoric and hermaphrodism, asexual versus sexual reproduction, the mechanisms, whether it's bet hedging or competition or the Red Queen hypothesis. And then when just on the way out at the end of the day, we're just going to talk about some generalized life history strategies for some generalized large categories of invertebrates, like as shown there. And so my goals is to think about costs and benefits for the invertebrates of sexual versus asexual reproduction, and to be able to interpret different examples when we're presented with them from the vast morass of invertebrate taxa, to think about and to contextualize what mechanisms might actually be driving the trends through evolutionary time towards the entrenchment of these strategies in different taxa, and how it might not be, in some cases, one taxa moving alone, but several taxa moving each other forward or along. And then thinking about the generalized strategies through time. So reproduction, we know this, and we talked about this. This was a slide. This is kind of picking up a thread from Monday. Reproduction is critical, and in the context of these energetic, essentially discussion-based equations that we're making, which is what I would make the analogy when we're talking about life history. Non-mathematically, we're still kind of invoking this large general calculus. So that second bullet there is survival is only relevant as it affects reproduction in this kind of very reductionist view, very reductionist calculus that we're doing here today. And the organism in this case is the unit of selection. That's the unit that selection acts upon, that little planular larva, spiraling through, settling down, and growing into a polypoid. Kin selection can be important, and we talked about coloniality on Monday, and where that might tip the balance and lead you to think that the selection is not just acting upon an individual, but acting upon a colony. And when we were talking last, and I'm just staring into the waiting room, sorry. We're talking about sexual, asexual, and sexual reproduction. I'd like us to kind of revisit that and think about from in an evolutionary perspective, in the evolution of metazoans on our planet, that we've got contemporarily, we have germ cells, right? We have our multicellular body reduced down to a single cell, or single cells in the case of sperm, or a single cell in case of eggs. So, and that germ cell lineage, is the phrasing that we've learned, is segregated early in development. And so sexual reproduction involves an energetically intensive return to a single cell stage. whereas asexual reproduction can form from propagules, that rather brutal knifing of the platyhelminths that we watched together on Monday. But I want to return to the protogametes of an early evolutionary. Let's go backwards in time to millions of years ago on the planet, where we have protogametes. And we have kind of two strategies that a protogamete would have to, or two problems they would have to answer. You want to fuse with the protogametes of another of the same species, another individual from the same species, and two, you want to carry sufficient resources to nourish whatever is going to be growing. And the problem in an evolutionary and in a life history context is that those two tasks demand opposing kinds of resources, opposing kinds of gametes. So what I'd like you to do, find your annotation tool, and we're going to look at this distribution. So it's a frequency histogram, so the frequency of a thing. And the x-axis is going to show you gamete size, so what I'd like you to do is to model the frequency of the gamete that would be competing for bumping into others of the same species versus, on the right-hand side, the zygote or the gamete that's going to provision. And what would those look like, both in terms of frequency and size? This is great. Okay. So we have two opinions with different frequencies associated with them. So we have a linear decline or a kind of a bimodal distribution. And so thank you for adding these hypotheses. Well done. I'm going to pull them off the screen. Actually, let's leave them on the screen for a second, because what I'd like to show you is there underneath yours, and I'll clean up the screen so you can see that better, is the idea that along the x-axis for gamete size, selection would tend in a pool of protogametes across a pool of individuals across a pool of a billion or many million, hundreds of millions years younger Earth. It probably, and it seems to have in our case, resulted in the selection for the gamete that competes for fusions is going to be numerous, energetically per individual, much less intensive, and small. So that's the protosperm here. Whereas the egg is going to be less frequent and much bigger. And this is essentially, sorry for bumping the microphone, this is then seen through the window, through the filter of a life history evolution, to think about the energy behind each one of these things, and being driven in this case by disruptive selection. So no one of these strategies is better than the other in this case. So disruptive selection then resulting in the occurrence of two equally optimal strategies that depend on each other to achieve a goal for one species. Now the thing is, if we look at the distribution in the world of sperm lengths, and we know a lot about sperm length around the animal kingdom and within invertebrates also, and this is what we're looking at. We're looking at your Dunedall phylogeny, so this capturing in a phylogenetic perspective of all of the different animal phyla. And then the yellow color map on the right-hand side, yellow to blue color map, is looking at the log distribution of sperm length. And it's logged because they cover orders of magnitude. So the prediction is in the anisogamy, or the evolution of different gamete types via disruptive selection, that sperm should be numerous and small. And so my question to you, and you can answer this on the screen or in the chat, does this figure this figure representing large contemporary meta-analyses that capture most of the data, does this support the prediction that sperm will be numerous and small, in particular small, because we're not looking at number, we're looking at size here. Throw your hypotheses in the chat or on the screen. I see an N on the screen, so it doesn't support it, and I see I see a yes question mark. So some qualitative support for yes, and some no question marks. This is good. Okay, so tell me about the question marks. All right. Okay, I'm going to pull the chat up here. So, yes, question mark. Since arthropods are a notable exception for their maximum size. Yeah, that's true. So, they are a notable exception. We've got some Drosophila whose sperm length is many times their body length, which is kind of ridiculous. No question mark, because most are shown in yellow. Yeah. So, tell me about the question marks, if there's anyone else, yeah, mostly mid-sized. Yep. And that's maybe a limitation of the color map, because when we have such a large scale change. But I would say yes, I think the question marks and the yes ink question mark, no question mark is actually a pretty good expression of whether it does. Yes, it does in most cases. So also it doesn't always. We see most taxa have small sperm. And if you were to overlay another color map on this, and you can kind of do this with your eyes because we've talked about this before, so I think you can kind of look at all of those tip names, all of those phyla, and say, who's mostly aquatic, who's marine or freshwater, and who is mostly throwing their gametes out into the water. And if you do that, if you kind of use your filter and flip that over your glasses or over your eyes, what you'll see is that most of the most animals or most taxa whose sperm is diluted, those are the short ones. If there's direct transfer, terrestrial-based direct transfer of the sperm, and then we start getting into the arthropods, into the insects, that's when sperm length seems to be released from this initial disruptive selective conditions of being small and numerous, and we see the evolution of longer but if you're a sperm caster, then you are, and externally fertilizing, then you tend to have smaller sperm, as would be predicted from, say, life history first principles. Okay, thank you for that. Now, what I'd like you to do for a second is just actually, and I was going to say, you can go here if you want, but you could also put it in the chat. Is there any other reason? What other factors can influence? So I talked about the environment, right? What other factors might influence sperm length? And if you're feeling like going to that Google Sheet, I can go and check. It's scannable. Or you can put it and surprise me with it in the chat. Oh, I see the chat showing up. All right, let's work there. Oh, that's interesting. Intraspecific competition invoked. Yeah. So the idea with male-male competition, so bigger sperm, if you've got a great big sperm, you're essentially using up all of the space, potentially in an internally fertilized environment, so there's less room for other male sperm. I think that's a great idea. Diet. Sure, because remember, we're talking about things in a life history, zero-sum salary cap kind of an environment. So if we're making less of any one of these germ cells, we're probably saving some energy that we might put into somatic growth or expression in some way. Oh, and Caden, that it might be correlated with egg size or with egg length or egg size. And the longer the journey, the more sperm are needed to make the trip, and the smaller the journey, the more of a sperm battle it has to be. I like that. Yeah, and I think, Gavin, that fits into the aquatic terrestrial split the environmental split that I was talking about. All right. Thank you. That's great. Oh, and I see your question, Morgan, in the chat. How can we say if they're small or not when we don't have a reference to compare them to? So we can talk about, well, you can see it in that distribution. The reference is the phylogenetic perspective of all of the taxa for which we have measurements. That'd be one. And the second would be you could do it as a ratio to body length or to body size. Okay. Now, I've been looking at the chat, so like Caramel, I think we don't have to wake her up here. She can keep sleeping in the sunshine, and we can keep going. And we can think about, so we've thought about the evolutionary underpinnings and selective pressures and costs or benefits underneath having two different gametes, the drivers behind it. And let's think about what pressures might involve having, I suppose, two sexes. Let's maybe dial it back even further and say, so what you've been taught probably in your career as a biologist is that sexual reproduction generates variation. So it's advantageous because it generates variation and that allows you to deal with a challenging environment. It's essentially rolling of the dice, scrambling and rolling of the dice that provides solutions to an abiotic environment, a competitive environment, or something that does not want you to survive and reproduce. But it's stressful to the individual because it involves this return to a single cell stage, which might be vulnerable, which might lead to the loss of good habitat or the loss of building upon historic investments in the soma. And so we see this distribution now of two different kind of strategies. Many animal taxa are not gonachoric, or don't have males and just males and females, that we see hermaphrodism, so that one individual has both. It's not asexually reproducing, but it contains the equipment of both sexes in one individual. So in gonachoric things, and we can see really, like many of you are familiar with this, the blanket octopus as an example. Essentially the sperm packet that's comprised of the male, so a 1-inch long male and then a 2-meter long female. The largest sex discrepancy, according to Wired magazine, in the animal kingdom. And I like their analogy, a full-size human female and a walnut-sized male. You just carry them, you could have like a little pouch you carry them around in. Maybe that's why there's never been pockets designed on women's pants, because they'd be worried about men being kind of sexually selected down to walnut size and just being carried around as the female blanket octopus does in a small pocket. So within a gonachoric population, there's male and female gametes assigned to separate individuals in the population. They're either reproductively male or female. That's their action exclusively. This is what you've been taught. There's only one kind of reproductive equipment. That's the benefit in terms of the investment. And then the cost is that you have to find a mate that has the opposite equipment of yours. We talked about a phylogenetic perspective. Here it is for an estimation from a large meta-analysis in the 20th century about the distribution or the frequency with which taxa with the animal phyla of the world demonstrate gonachoric from a mostly or all to a hardly or ever, mostly or all at the top of one, a darker scale, to a hardly or ever at the bottom to a lighter scale. And you can see its distribution widely across some of the diverse ecdysozoans here. We see it widely, but not exclusively, across the deuterostomes and intermittently across the spiralians. And I want to push back a little bit on the idea that this is just a gonachoric male or female. This is a two-tiered kind of a thing. And I want to introduce the idea that in all levels of biology, this is much more of a gradient than people in the 20th century and that perhaps you, as a student in the early 21st century, were taught. And I'm going to talk about this example with tadpole shrimp, which is a life cycle that's demonstrated here. So tadpole shrimp breed in ephemeral environments, often in the Midwest of North America or Australia. That's a broad kind of a distribution. It's not just one species. But this taxon, which was traditionally categorized because of our biases as males or females based on whether or not there was morphological dimorphism. And so they would see these little Napoli larvae that would develop into two different things, and we called them males or females. There's the male on the bottom. This is what would have been called the female in the past. But female here is actually a functional hermaphrodite. And the hermaphrodites are really critical to this species that lives aquatically, but in very ephemeral and dry, temporally dry environments, because they create and bury desiccation-resistant eggs in the soil. And so we see some outcrossing that can happen between hermaphrodites and males, and then we see the production of these eggs by a selfing within the hermaphrodites. Those then, the resting eggs, drift down into the soil and can wait out. It's essentially a diapause, as a phrase that's more frequently applied to in plant or botany circles. So they wait out that dry, and when the water returns, they can hatch rapidly under spring and summer conditions when the rain happens, and then they can grow rapidly, move through the Napoli stage, and produce hermaphrodites. And so I see the question in the chat because I glanced down. So yes, and this is one of the costs of being a hermaphroditic population, is selfing with yourself. Potentially is problematic, but it also means that you can bump into anybody can bump into anybody and have a functional reproductive pathway laid out in front of them. But selfing is a way to get eggs that can let you escape, right? So it might not be advantageous to mate with yourself, but if it's going to produce eggs, and the eggs is the only part of your life history stage that's going to survive the drying that's coming, this is advantageous. And so when we look at the tadpole shrimp in a phylogenetic perspective, and so like I said, many species from around the world, what we're looking at is in blue, where the systems of tadpole shrimp tend to be strictly gonochoric. Orange is not just hermaphroditic, but it's this co-occurrence, which is called androdiacy, where there is a polymorphism in the population of both males and hermaphrodites. And the idea is that it suggests from an evolutionary perspective that there might be, this might represent a transition in a long-term, in evolutionary time, a transition from gonochoric to being hermaphroditic, strictly or obligately, I suppose. And that, and it's likely due to this selection for reproductive assurance. So it lets you deal with potentially low density, because anyone can run into anyone else and they can mate, or limited dispersal, because you're a shrimp, a little crustacean in a pond. You're not going to be able to walk long distances to the next water source, so you need to escape. Instead of dispersing through space to escape, you need to disperse through time. So just like Doctor Who has his time and relative dimension in space machine, these are the eggs, right? They get you out of the problem by letting you wait out whatever the stressful event is. So moving from thinking about gonochoric to hermaphroditic individuals. So we've got, in a hermaphroditic, truly hermaphroditic individual, we have the male and female gametes produced within one individual. So a cost being, as I've mentioned, you then have to, the individual, the organism, has to produce and possess and build two different kinds of reproductive equipment. The benefit, that's costly. The benefit is you can mate with any other organism of the same species that you run into. Simultaneous hermaphrodites and sequential. So this is two different terms that relate to when the male and female gametes are produced. If they're simultaneous, it means you're building them and you're possessing them at the same time. Now the cost that Gavin's comment made me think about, sequential hermaphrodism is a cost that might reduce, or is a strategy that reduce the likelihood of selfing, of mating with yourself, because the sex reversal happens in the life cycle. So we've got an example with Crepidulae fornicate, the slipper snails, where they are sequentially hermaphrodite. So they start out as males and they finish as females, I believe. The order doesn't matter in this case. And here's what we would look at for the distribution, again, in a back of the napkin kind of a calculation from a late 20th century review, where an eminent scholar was asked to think about all of the phyla of the world and say, well, how frequently do we see this in taxa of each one of these phyla? And you can see a much, broader distribution of hermaphroditic because we've got all sorts of the non-bilaterally symmetrical lineages that demonstrate it. And the benefit for seems to come about largely in dilute populations where it's hard to bump into one of yourself, or ephemeral populations where the conditions are going to be quite dramatically stressful, and the need to escape them in space or time, particularly time, is strong. Or sluggish, where you might be around, but it's like who was Jenny Slate's snail character? It's really hard to think about, racing across to have a Romeo and Juliet. Romeo and Juliet has hardly ever been cast with snails or platyhemionth flatworms. So sluggish animals don't need Romeo or Juliet. They are Romeo and Juliet. The cost, of course, then being energetic cost, as we've talked about, and accidental self-fertilization. So the cost of sexual reproduction that we've talked about, so finding a mate, there can be all sorts of danger to yourself in terms of investing somatically in kind of crazy display ornamentation, in courtship or fighting, that maybe any or all of those features or phenotypes leave you with an increased rate of risk of predation, the overproduction of gametes, lots and lots of potential investment that doesn't, if you're not successful, if there's not actually success, not just attempted reproduction, but successful reproduction, you've wasted kind of all that energetic input. Most offspring do not survive. And then timing the gamete release. What kind of signatures do you use? Are they abiotic signatures? Do those abiotic signatures change through time? This is, of course, where we would tie some of this life history theory at the start of this course into some of the discussion about the climate crisis and diversity crisis at the end, because the timing is often, we are in the Anthropocene living through a world where the signatures, the abiotic signatures that historically would have allowed the bearers of different gametes to organize and time the synchronization are falling out of whack. So let's talk about the underlying underpinning mechanisms that are likely behind the appearance of sex, because sex has benefits, sure, but variation, blah, blah, blah. But it also has a lot of costs. And so we shouldn't expect to see it, I would argue, unless there are some benefits. And there are three expressions that we're going to kind of work through, three mechanisms that let you think about those benefits through time. The first of them is this bet hedging hypothesis. And this draws on the example that we were just thinking about expressed for hermaphrodites. So two examples here. On the left-hand side, we have Daphnia. Daphnia, one of the most numerous, like as you've been out in the Arboretum, there's tons of Daphnia in those ponds if they're still standing, but even in some of the deep water ponds that you've been walking around where there is certainly Daphnia still living and floating around, there's also lots of ephemeral ponds, Goosewoods Walk, that probably are dry this year, but there are Daphnia there as well. And what they've done is that they have they've fallen out of the water column and they are surviving as eggs via sex. So otherwise, they're reproducing parthenogenetically, and that's fast and it allows for rapid population growth. But sex occasionally, coincident with the oncoming dry or the oncoming winter or something stressful, allows you to escape that stressful event. And so this is an expression, we think, of some of the benefits of sex. Dolid rotifers, so environments that are prone to desiccation or other kind of environmental fluctuations, they live asexually, and they're very common in these kind of unpredictable and stable environments. And so they can reproduce quickly without needing mates that allows them to colonize unpredictable environments rapidly. Now, what about even within the same species? So kind of building on this Daphne example, let's move into some, still within the hexapods, within the arthropods, thinking about these aphids. Aphids, that this could be any of the stems of burdock that you're walking by on your way into the school, that will be probably tended by ants at this time of year, eating some of the phloem, some of the juicy, sugary water that they're secreting at the tail end of their at the end of their alimentary canal. Aphids commonly reproduce asexually when food is abundant. They have rapid reproduction, just like the Daphnia. When the environment becomes saturated, they switch to sexual reproduction. What does that look like? So in the spring, things are growing, things are fresh, there's like the plants are returning, there's photosynthesis, and we've got leaves and drawing water up and just a whole bunch of resources. They are asexually. Parthenogenic females are predominant in spring and early summer. But in the fall, when the environment tends to become saturated with a lot of aphids and the proportion of resources is going down and being more tightly partitioned, we see this switch to sexuals that produce eggs that can overwinter. So asexual reproduction being fast, taking advantage of a new unsaturated environment. And then in a saturated environment, sexual reproduction can give organisms an example to get through that stage. So bed hedging, what's behind sex? Bed hedging, competition, and then something you've been introduced to before, certainly, which is this Lewis Carroll analogy, the Red Queen hypothesis, that you need to run as fast as you can just to stay in a place, meaning that in this biological analogy, that there are other taxa. If you can't kind of give up this race or be comfortable, lay back, sit back on your laurels because your predator or your parasite is going to consume you very, very rapidly. And the idea here is that in a why does sex exist? Perhaps it's an expression of the Red Queen hypothesis. So that sexual reproduction gives you short-term advantage over competitors that are only using asexual reproduction because of the differential advantage it gives you against predators or prey. Predators or parasites. And there's an example of that, a famous one, with this snail, Potamopricus antipodarum, which sounds very spell-like, Wingardia Leviosa. That's the snail. And if you don't want to say Potamopricus antipodarum, you can say the New Zealand mud snail. So this is a freshwater snail that's native to New Zealand that is characterized by the same kind of androdiasis, so reproductive polymorphism, where there are both obligately sexual individuals and obligately asexual individuals that coexist in populations around New Zealand. And what we tend to see is the role of this organism over here. This is a whole other phylum. This is a trematode. Okay, so this is a parasitic, so a fluke, and this parasitic worm, whenever we see an increase, and so kind of do your squinting your line of best fit here and say where would that, we would see basically an increase like this, that the increase in the rate of parasitism, an increase in the frequency of the trematodes results in an increase frequency of males, i.e. sex. And so this is frequently the Japanese mud snail, and it's the effect upon its reproductive biology of the trematode parasite is seen as an expression of the Red Queen hypothesis underpinning this particular entrenchment of sexual reproduction. All right. How does sex versus asexual reproduction give an advantage over predators and parasites? It essentially comes down to the rescrambling. That if you've got your asexuals that are out reproducing fast, but always the same, and those are prey uponable by the parasite or consumable by the predator, then the idea, the hypothesis, the that critical part under the hood of the mechanism is that sex must give you a way to escape. So it must essentially, like if the predator or the parasite is coming in through an evolutionary open door, some part of your behavior or life history or your physiology that allows you to be preyed upon, the idea would be that sex eventually would kind of stumble upon a solution where that door is closed and those individuals would have an advantage in this kind of a population. Does that make sense? Good question. So yeah, so that this is kind of similar to that tadpole shrimpy thing. Now I want to pull these back in our final few minutes here, thinking about life history strategies and thinking about. I've given you several analogies. There's my favorite one about pie. There's my second favorite one about salary caps. This is another kind of a diagram that is often used when modeling or when thinking about life history to think about a Y diagram. So you've got your essentially some expression that in this Y model of resource acquisition, this trade-off, that whatever, so you've got you going through life, this organism, this tadpole shrimp, this trematode, this snail, mud snail in New Zealand, this kind of hobbit-based mud snail going through the aquatic environment in New Zealand, acquiring resources, and where it can spend those resources can be divided into investment into survival or fecundity. And whatever goes towards survival can't be put towards fecundity and vice versa. So this is that dichotomy affects the survival at the expense of the other path. So the idea that every organism's life history is some kind of evolutionary compromise. So look at us. We're talking about gradients and not binaries. We're talking about compromises. We are an informed bunch of biologists. But how informed are we? Well, actually, about most of the taxa, not so much. This is one of the most recent, this is kind of my re-expression of a very proudly abundant meta-analysis into complex marine life history strategies, where they synthesized like 3,315 different examples to talk about marine life history strategies, successful and abundant marine life history strategies. So that's a victory. Pulling all those studies together, that's a victory. But also, one of the things that doing a meta-analysis lets you do is to think about opportunities. Some people would talk about them as problems that you would solve. Let's talk about opportunities. The opportunities I see is that right now, in such a meta-analysis, which is a concentration of all the scientific literature, we see only 3 phyla: the arthropods, the annelids, and the mollusks. Strike the annelids. It's the echinoderms, the mollusks, and the arthropods. Contains 70% of those species. And so that's the red bar here distributed. And you see it mirrors a lot of where our favorite things are. So perhaps it's an expression of some of our biases towards things that are larger, things that we enjoy looking at and working with. But it's also then an expression of where we need work to be done in the future. That's where you come in. I'm going to mute for a second and cough. All right, I apologize. I hope none of that crept through. Final few minutes. Let's talk about some generalizations, some generalized terms, some generalized distribution of traits across large habitats, marine big things, marine small things, terrestrial things. So when we talk about these different life history traits, remember back at the start of Monday we talked about different traits that would fall into the menu of someone interested in studying life history traits. Here's a lot of them. Now you can see it's a simple list, but if we were to examine them across, more muting. across a sexual divide or a gonochoric divide, we can see there's a huge number of combinations, but that some of the trends that we've talked about are highly correlated. So lots of traits across lots of taxa, across gonochoric things, lots of combinations, but lots of correlation. And that cycles back to some of what we were talking about in terms of thinking about fast or slow things. That's where a lot of the life history research tended to go in the start of this century and the tail end of the last one. In our examination so far of the invertebrates, we've been focusing on marine systems a lot. And so how young get nutrition, whether they're planktotrophic or lectotrophic. These are life history strategy. This is life history informed strategy, that the planktotrophic things, the larvae that feed in the plankton, allows for longer dispersal compared to direct development and generally compared to lectotrophic development, because the yolk runs out and you tend to fall out of the water column. And there's even a third category, matrotrophic embryos. So these are ones that have even a greater investment from the parent. And we see it with some bryozoans, some zooarthropods, and some platyhelminth flatworms. So this includes more than just a yolk. It includes incubation chambers or brooding or nutrient secretions in the uterus or other incubating spaces, or actually the consumption of maternal tissues, other eggs, or sibs. So sibs that are born so that only some of those eggs, but their role is to be consumed by others. So relatively low maternal investment, long planktonic period, wide dispersal, but high mortality. On the other end of a continuum, again with the continuum, we see that they're not feeding because they have these high maternal investments. Larvae, shorter planktonic period, limited dispersal, and reduced mortality. So generally in the marine environment, we see eggs discharged into water. We see external fertilization. We should see small yolks with small eggs with small amounts of yolk. We tend to see very small sperm. We see planktotrophic larvae formed that can widely disperse. Reproduction that has tight ties with seasonality. So potential vulnerabilities in the Anthropocene. That's in the small marine life. In larger marine life, like we're not talking like blue whale leg, but big, but ketognaths up, the eggs are often brooded. So lectotrophic versus matrotrophic eggs. Internal fertilization, frequent reproduction throughout the year with a less death in the larval stage. In the freshwater and the terrestrial environment, we see a whole bunch more investment in getting your young through, because we see viviparous things and the use of impermeable eggs or cocoons. You remember with the isopods, the crustacean, one of the most successful terrestrial crustacean lineages, they use the marsupium, so they carry nestled amongst some of their coxa, the first segments, the first articulation of their legs. They have these adaptations in their coxa where they can grow and keep moist their young in a terrestrial environments. They have complex internal fertilization, longer sperm, larger eggs. We see brood care and provisioning, episodic egg laying, and some production of hermaphrodites, and the idea of diapause, escaping not just dispersing, not flying away, but going down, hunkering down and waiting something out in space. Okay, glance over at the chat. I don't see anything new here. And so come back over and think about a couple of other, we've talked about semilparous and iteroparous, so you've run into these things, particularly if you've been dealing with fish, any courses that have talked about fish, because this is famously where we talk about fish that have one reproductive event with a maximum amount of resources than death, or iteroparous things that reproduce a few young at one time and then reproduce throughout their lives. And so we see amongst marine mollusks, we see squids and cuttlefish and octopus and kind of everything except for the nautilus were for a long time considered to be semilparous. But they actually, the more we learn about them, the more we learn that there's variation with complicated reproductive behaviors. So we see some, like the minimus, the small octopus, where all oocytes mature at the same time, so they're synchronous and they're semilparous. But then there are others where the oocytes don't mature at the same time, and instead all stages are present in the ovary at the same time. So they're asynchronous and iteroparous. So even the things that we thought we knew, we didn't. And this lets me cycle back right at the very end to thinking about, this is that this is their phylogenetic expression of their large meta-analysis of another meta-analysis that shapes, so life history traits shaping the diversity of the world. And this was particularly their effect, or their review was on body size, reproductive mode, and also on longevity. So one of the kind of axes of life history theory that we haven't talked a lot about. So in this case, they got nearly 1,000 studies, so over 800 studies. Only 44 of them were of invertebrate species that had sufficient information to be included. And so I say this again to you as an expression of the fact that there's a lot of this when we think about these large evolutionary rules, evolutionary norms, evolutionary expectations. We also kind of coincidentally ought to be thinking about them as biases, because the biases of our experience and the biases of our phylogenetic lineage, the deuterosomes, where we tend to see things the way that we are, most of life doesn't exist the way that we do. And so there is much work to be done. All right. Final check-in at the chat. I don't see anything new, I don't think there. So let's just wrap it up then and say, in the second kind of swing through or slice into the pie, we've talked about sexual reproduction, the evolution of two gametes, the entrenchment or the continuum of gonochoric to hermaphroditic, the benefits or costs being associated with being sexual or asexual, mechanisms that might underlie sexual reproduction, and then some generalized life history forms and strategies across the world, with an invocation that much remains to be learned. There is much work for you to do. And where are we going? So lecture next week, we're going to continue some of the reproductive theme. We're going to talk about mating and molting and respiration. So the first part of the week will kind of extend some of what we've talked about now. When we start talking about molting and respiration, we're going to talk a lot about the external body wall and chitin and how you interact with the world. What to be working on. You've got your invertebrate R videos, and those are due, it's like weeks away, but it's one of these month-long experiments that comes up quickly. So if you haven't started working on it, I would suggest getting there and beginning, and giving yourself time to think about the product at the end, because the end and what's being marked is, of course, a video. Your second case study is going to be released Thursday morning, and you've got from Thursday to Monday morning to do that. And then your, it says, this is a mistake, I believe. So your class experiments, you can start reading about them, but I confirm this with your schedule and with Shayla, but I believe you don't have a lab in person next week. So that's it. This is the end of Wednesday. I hope that you have some capacity to get out and enjoy and be invigorated by the sun and the summer-like conditions as they kind of go away, I'm told, at the end of the week. Take care of yourself. Take care of others around you. And I'm going to stop sharing and hang around for a few minutes if there are any questions. So